Oh, hey, Nico. I didn't see you there. What are you up to?
Oh, I'm working on my lab reports.
Oh, the fly one? Me too. I have to add the finishing touches on mine now.
And the yeast one.
Wait, that was due like a month ago.
Oh, I know. I just never understood what a complementation test was, so I just didn't do it.
For a month?
Well, I didn't do any of the readings, and I slept through the debriefs.
Don't worry, I can try and explain it to you right now.
Really? You would do that for me?
Don't be weird about it. Essentially, complementation tests are used to determine whether two mutations that cause the same mutant phenotype affect the same gene or separate genes. When geneticists collect a lot of mutations affecting a specific process of interest, they first need to determine how many different genes their collection of mutations affect.
What does that have to do with yeast? I thought yeast only matters when you're baking bread.
Well, yeast are useful as a model organism because they exist as haploid or diploid organisms. If crossing two yeast haploids that each display a mutant phenotype produces a diploid with a wild type phenotype, the mutations affect separate genes. They complement each other. If the cross yields a mutant phenotype, the mutations affect the same gene. The diploid now has two mutated alleles for this gene. This is all assuming the mutation in question is recessive, of course.
How the heck does that work?
Well, when two yeasts with mutations affecting separate genes are crossed, their offspring will inherit one wild-type copy of each of the genes, producing the wild-type phenotype. When two yeasts with mutations affecting the same gene are crossed, their offspring will not inherit a wild-type copy of the gene, as it is inheriting one mutated copy of each gene from each haploid parent, and the mutant phenotype will be observed.
Girl, I'm lost.
Look. If we cross the HA1 and HB1 mutants, the offspring will be a mutant because both mutations are in the AD1 gene. But if we cross the HA1 and HB2 mutants, the offspring will be white because they are heterozygous at both the AD1 and AD2 loci. You follow?
Whoa, did we just enter your phone?
Can you please log in?
But I'm not yeast. Why should I care about any of this in the first place?
In this case, we're using yeast as a model organism, but these concepts can be applied to other organisms too. Complementation tests are extremely useful in establishing pedigrees, mapping genes, and determining the genetic basis for many phenotypes.
Funny, some people call me a model organism.
Please stop. If two mutants produce a wild-type offspring, we say the genes complement each other and are in different complementation groups. If they produce a mutant offspring, then the genes are in the same complementation group and they fail to complement each other.
That is so rude. I love compliments.
Okay, I need to go. Do you think you understand complementation tests now?
Can you say that one more time? I need to write into my lab report.
Bye.
Wait, no, come back.